In this part, we'll be discussing about the female cones of the Pinus. Female cones are produced again in clusters. Now this is a long shoot. On the long shoot, we can see in the axil of the scaly leaf again, as we saw in case of male cones. In the axil, these are the scaly leaves. This is a long shoot. These structures are scaly leaves. In the axil of the scaly leaf, what is going to arise? This strobile or a female cone. Sorry. The female cones are produced usually in pairs or in clusters in the axil of the scaly leaf. So here we have a single female cone, but this picture here we can see a clusters of some 3 female cones. So they are usually produced in pairs or in clusters, or even the chances are there that they may be even single in nature. The female cones, they mature very, very slowly. That is, maturation period is like of 3 years. At the end of a 3rd year, a female cone will be mature enough. That's the reason we said during the development of the male cone that it is a protandrous in nature, in the sense the male cones are going to develop first. Later the female, which grows very slowly with a duration of some 3 years. So we are going to see here different years female cone. So the first the female cones, as I said, they mature very slow. The first year young cone is about like 1 to 2 centimeter in length. It will be that small. It is soft in nature. So this is the picture of the first year female cone, which is usually 1 to 2 centimeter in length. It is soft in nature and it is compact, usually green in color. To make it more attractive, this is the first year cone, which is green in color. Usually this is an enlarged view, but they will be like 1 to 2 centimeter in length. They are compact and green in color. Whereas the second year cone is like comparatively large. It is like 5 to 8 centimeter in length. This one, this upper one, not this brown color, okay. These 2 cones, these are of the second year cone, which are compactly large. As we saw in previous picture, green compactly arranged, that was the first year cone. This is the second year cone, which is compact only, but it is comparatively larger. It is like 5 to 8 centimeter in length. It is not soft. This change, like it is going to change from the soft to woody now. It is woody in nature, not at all soft. It is compact, and even the color is changing from green to slightly orange in color. And the fully matured 3rd year cone, this is the 3rd year cone, the 3rd year cone is fully matured, and it is much more larger in size, about like 60 centimeter in length. It is woody, complete woody, and it is not compact. We can compare this and this. This is completely compact, where this is loosely arranged, and it is like brown in color. Here the megasporophylls are separated from each other. Why this separation? Reason behind, we have this central axis. This is the longitudinal section of the female strobile. This central axis, it is going to elongate. Elongation of this central axis, udduk beliyudu. Elongation of this central axis lead to separation of these megasporophylls. This occurs in the 3rd year, that is during the fully maturation stage. Now the female cones of the Pinus represent what? A compound structure. Usually the female cone is composed of the central axis. When we are going to go for the longitudinal section, it usually consists of a central axis. On the either side of the central axis, what we have some 80 to 90 megasporophylls. So these are what? These are nothing but megasporophylls, which are arranged spirally. First year, second year, 3rd year. This is the black and white structure. This is the colorful one. This is the first year structure, female cones. This is the 3rd year one. See how the change is from compact green soft first year to completely matured 3rd year, where we are having this woody, loosely arranged and brown in color cones. This is somewhat of second year cone. From green it is changing to orange color, and also its length is bit lengthy compared to the first year. Here this is like second and third year. Yeah. Now speaking about the longitudinal section, as we have told, on the either side there will be megasporophylls arranged, say about some 80 to 90 in number, and usually the apex of the megasporophyll and the basal region won't have these megasporophylls won't have microsporangia in them. That's the reason these both upper as well as the basal region of the cone are sterile in nature. They won't bear any kind of spores. Now a single megasporophyll consists of two scales. Single megasporophyll, when we are going to focus on, it usually consists of two scales. One is ovuliferous scale. This one will be the broader one. The broader one is ovuliferous scale, and the small membranous structure, this is bract scale. So a large woody ovuliferous scale, these ovuliferous scale bear ovules. So these are the ovules. These round structures are the ovules. Where the ovules are present? On this scale. That scale is called as what? Ovuliferous scale. We have two types of scale. One is ovuliferous, another one is bract. Ovuliferous is somewhat woody and large compared to that of bract scale. This ovuliferous scale usually consists of ovule, whereas the bract scale is like membranous, brown in color, and it is usually present on the ventral surface, whereas this ovuliferous scale is present on the dorsal surface, like this. Ovuliferous, bract. Here we have ovule, ovuliferous scale, bract scale. Now the ovuliferous scale is a thick, it is large, yes, and it is woody in nature, and it is somewhat triangular in shape. Likewise, at the tip we have this triangular shape, and it is brownish in color. Its upper thick exposed part, this one, is usually called as apophysis. Is usually known as apophysis, which is nothing but it is going to function like a lid or a covering. In the mature cone, the tip of this apophysis becomes the umbo. Now here, this in the mature one, this apophysis becomes umbo. Now what is umbo? Umbo is nothing but have you seen any this shell animals like snail and all? There on their outer part we have this shield, thick covering of calcareous shell. On this calcareous shell we can see this like a rounded structure, circular concentric rounded structure. This is nothing but we are going to consider it as an umbo. That is a shield, a rounded elevation, nothing but a kind of a covering. The same kind of an covering is formed by this apophysis. There it resembles the umbo of those shell animals. That's the reason here also they have named it as an umbo. It is nothing but a covering. So in the mature cone, at the tip of the apophysis, this tip of apophysis becomes the umbo, and there is a supply of two separate vascular traces. One supply goes to the ovuliferous scale, another one supply goes to the bract scale. Now the development of microsporangium, that is development of megasporangium, that is ovule, is of the eusporangiate type. An ovule develops from the group of cells, superficial cells of the ovuliferous scale. That is, on the ovuliferous scale, development of the ovule takes place. The development of the ovule, or nothing but a megasporangium, is again of eusporangiate type. In the sense, a group of cells from the ovuliferous scale are going to divide and redivide, leading to formation of what is called as an ovule. So this was a description regarding the female cone. Thank you.